Toxicology is a form of pathology in the sense that pathology looks for the cause of disease, but where pathology involves the examination of tissues, organs, and bodily fluids in order to study and diagnose disease, toxicology examines those samples to discover the presence of chemicals which can have an effect on the health of the patient. Most toxicology testing is looking for chemicals which are foreign to the human body, though some toxicology tests may seek to gather data about naturally occurring chemicals, like hormones, such as insulin. Another distinction is that pathology can include microbiology, which looks to identify microbes, whereas a toxicology test will not seek to identify species of bacteria or fungus. However, a toxicology test may still indicate the presence of a microbial infection because some microbes produce toxins which may show up on a toxicology test. Toxicology tests are looking to use known chemical properties to identify the presence of certain relevant chemicals related to medicine and drugs of various kinds. The purpose is to give evidence to physicians of what drugs their patients have or have not been taking. Sometimes toxicology seeks to prove that an individual has been ingesting illegal drugs. Sometimes it is merely to determine if a patient has been taking their prescription. For any toxicology test, the chemical substance that is the subject of the analysis is referred to as an analyte. An analyte may be a parent drug or a metabolite. When a physician sends a patient's urine sample to be analyzed in the toxicology lab, the sample is also tested by the clinical lab to ensure it is a human urine sample. This is called validity testing, because it is seeking to determine that the sample is a valid human urine sample and not some other substance. This is especially important when the patient knows they are being tested for an illegal substance, because they may try to pass off another substance as their urine sample. Synthetic urine, which was used by drug testing laboratories for quality control, is now commercially sold and can be used to fool a positive drug test. When a sample is received, it is tested in the toxicology lab to find evidence of those analytes specifically requested by the physician. To this end, there are two different kinds of tests which are done: a presumptive screening test and a confirmatory or definitive test. Presumptive screening, also called presumptive testing or a drug screen, is a qualitative technique to identify certain targeted drugs. These tests are common in work or athletic environments because they can yield rapid results. The presumptive screen is qualitative, meaning it returns a result of positive, the analyte is present in the sample, or negative, it is not present. It indicates the presence of certain analytes, but doesn't provide an amount. The screen tests are inexpensive and are available for a wide variety of drugs, but they are not very precise. A screening result commonly indicates that a class of drugs is positive, not necessarily which specific drug triggered the positive. For example, the antidepressant drug sertraline, also known as Zoloft, can produce a false positive result for the drug class benzodiazepines, a class that includes both legal prescription drugs and illegal street drugs. Another disadvantage with screening tests is they are more likely to give false test reports due to a lack of sensitivity and specificity. A false positive occurs when the screen is positive but the drug is not actually present, and a false negative occurs when the drug is actually present but the screen is negative. The false negatives are believed to be commonly caused by the lack of sensitivity in the quick screens, as they don't have the ability to detect amounts as low as the amounts detectable by definitive tests. Confirmatory or definitive tests are more specific and allow for the detection of specific drugs or metabolites of interest. In most cases, presumptive testing is used because it is quick, fairly accurate, and easily accessible in a wide variety of settings. Definitive testing may be needed when presumptive results alone are not sufficient to guide clinical care. Definitive drug testing can also be used to rule out false positives or false negative results, as these tests are both more specific and more sensitive. A definitive test seeks to confirm the result of the presumptive screen with additional information. A definitive test results in a positive or negative with quantities for the positives, listed as nanograms per milliliter. This is the concentration of the targeted analyte found in the sample. These tests take more time and are frequently more expensive than the screens, but the results are far more specific, providing clear indication of what is in the sample and in what amounts. As toxicology is looking for evidence of the presence of certain analytes, the tests need to be able to identify many different chemical substances and tell them apart. Some tests commonly used by toxicology laboratories are immunoassays, chromatography, mass spectrometry, and spectroscopy. An assay is a process of analyzing a substance to determine its composition or quality. An immunoassay is such a process that involves a part of the human immune system, which are called antibodies. All immunoassays are based on the interaction of a target molecule, called an antigen, with a corresponding antibody. An antibody is a large Y-shaped protein used by the human immune system to identify and neutralize foreign objects, such as bacteria and viruses. The antibody binds to a unique molecule which is referred to as an antigen. Each tip of the Y of the antibody contains a binding site that is specific for one particular antigen, allowing these two structures to bind together with precision. This also keeps it from binding with other molecules, allowing a high degree of certainty for the identity of the target which is able to bind with the antibody. When applied to drug testing, the immunoassay technique uses an antibody specific for the drug or drug class being assayed. The antibodies are labeled by binding them to a molecule which can be identified, such as a radioisotope, which is an unstable form of an element, what we call radioactive. The sample to be identified is combined with these labeled antibodies and treated to isolate them so that the test is gathering those antibodies which have successfully bound to the target antigens and eliminating those which have not. The remaining amount is tested to measure its radioactivity, which gives an indication of how many antibodies successfully binded to an antigen. An immunoassay which measures the radioactivity of these isotope labels is called radioimmunoassay, or RIA, and it is popular because of its high sensitivity and its ability to analyze large numbers of samples rapidly. Chromatography is a laboratory technique for the separation of a mixture into its chemical components. As any particular sample to be analyzed may be made of many different substances, separating them is the first step toward identifying them individually. There are several different kinds of chromatography tests, including liquid and gas chromatography tests. The liquid in liquid chromatography refers to the fact that the sample in the test is carried by a liquid through the machinery, as opposed to being vaporized and carried in a gas, which is what happens at the start of gas chromatography. Some sources will say that gas is a quicker method, whereas others claim liquid is faster at producing results. Some sources claim that liquid allows a wider range of test analytes. Which test is preferred most likely comes down to the kind of testing which is needed. Testing gas samples would probably be more efficient in a gas chromatography test, where the primarily liquid samples provided by medical labs are almost certainly better served by a liquid chromatography test. Both are reliable methods of separating compounds out into their individual components so that those components can be analyzed. Mass spectrometry, also referred to as mass spec, is an analytical tool useful for measuring the mass-to-charge ratio of one or more molecules present in a sample. These measurements can often be used to calculate the exact molecular weight of the sample components as well, which makes it possible to identify them. Mass spectrometry identifies the masses of the atoms or molecules in the sample, as well as their electrical charge, and produces a graph plotting these measurements against each other. Knowing the mass and charge of a substance allows it to be accurately identified. As a crude metaphor, imagine you have two unlabeled boxes, one containing sweaters and the other containing books. By lifting them, you can tell which is which, because the box of books will be significantly heavier. The way mass spec identifies molecules is a little like this. Sample molecules are ionized using various methods. This produces charged ions, which are atoms or molecules with a positive or negative charge. If they have more protons than electrons, they are positively charged. A positively charged ion is also called a cation. If they have more electrons than protons, they have a negative charge. These are also known as anions. Mass spec uses a stream of high-energy electrons to knock electrons off of the sample, producing positively charged cations. These ions are then sent through the machine. Mass spectrometers detect the speed at which positively charged ions move through a vacuum chamber toward a negatively charged plate. The speed of the ions is determined by their mass. As the ions move through the chamber, the mass analyzer records the speed and relative abundance of ions to produce a visual readout, or mass spectrum. Different compounds in the sample will have a different mass. The readout enables researchers to determine the composition of the sample by comparing the results to known elements and molecular compounds. Combining the physical separation capabilities of liquid chromatography with the mass analysis capabilities of mass spectrometry is a method called liquid chromatography mass spectrometry, sometimes abbreviated LCMS. If instead the separation is done with gas, it is called gas chromatography mass spectrometry and abbreviated GCMS. These techniques allow complex mixtures, like those found in any biological sample, to be separated out into its basic components, and then those components are accurately identified according to their unique chemical and physical properties. It is a fast and accurate method of identifying the various substances in a sample. Spectrometry is different than spectroscopy, though both can be used to identify chemical substances. Where mass spectrometry, also called mass spec, is an analytical tool for measuring the mass-to-charge ratio of one or more molecules present in a sample, spectroscopy is the study of how matter absorbs and emits light and other radiation. The end of the word is similar to the end of microscope or telescope, which are both instruments for gathering light so that something can be seen. Spectroscopy is a process of using light or other frequencies of the electromagnetic spectrum to identify unique chemicals. Spectroscopy can be used to detect, determine, and quantify the chemical composition of a sample. Each type of molecule and atom will reflect, absorb, or emit electromagnetic radiation in its own characteristic way. Spectroscopy uses these characteristics to analyze the composition of a sample. To put this in terms which are more familiar, a white shirt and a black shirt will both reflect and absorb light energy when you're outside on a sunny day. The white shirt will reflect more light energy and therefore have a lower temperature, whereas the black shirt will absorb more light energy and have a higher temperature. This difference in reflecting or absorbing light energy is the principle on which spectroscopy works to identify different chemicals. Some labs will prefer these methods because they are non-destructive, meaning that the sample will not be chemically altered by the test. Other kinds of testing, such as gas chromatography mass spectrometry, are destructive because the sample is vaporized at the start of the chromatography and altered. For forensic tests, where the data is needed for a legal case, and the amount of substance to be sampled is small and there's no ability to gather more, a non-destructive method is preferred because it allows more testing with less substance. For medical tests, especially non-emergency testing, other methods are acceptable because it is always possible to acquire another sample if additional testing is needed. Isomers are molecules with identical molecular formulas, that is, the same number of atoms of each element, but distinct arrangements of atoms in space. These two different substances would have the same chemical formula. For example, glucose and fructose are isomers because they have an identical molecular formula, C6H12O6. Isomers may have different arrangements of atoms such that each isomer is distinct in its arrangement of atoms, or they may be mirror images of each other's structure, the way that your right and left hands are. Just as your right or left hands are each better suited to wear a specific glove, isomers do not necessarily share chemical or physical properties. They have the same chemical formula and therefore the same mass, but they may have importantly different effects on the human body when ingested. And so, distinguishing between them can have important legal and medical ramifications. In some cases, the differences are minor, such as the differences between glucose and fructose. These isomer differences can be found in medication as well. The isomers of ibuprofen are identified by the prefixes R- and S+. They are similar in properties such as melting point, boiling point, and solubility. However, the S+ form is used as an anti-inflammatory drug, while the R- form has no anti-inflammatory effect. Oddly, the R- form is converted into the S+ form by enzymes in the human body, and so ibuprofen is often manufactured as the R- form, which is easier and less expensive to produce. Methamphetamine is a Schedule II stimulant under the Controlled Substances Act, which means that it has a high potential for abuse and a currently accepted medical use in FDA-approved products. Methamphetamine comes in two isomer versions, the dextro or D-methamphetamine, D isomer, and the levo or L-methamphetamine, L isomer. The D isomer has a much stronger impact on the emotional state of the person ingesting it, as well as a much higher potential for addiction. This is why each isomer is treated differently under the law, as well as in medical applications. The dextro or D isomer is a central nervous system stimulant commonly known as meth or speed, and is a DEA Schedule II controlled drug. However, it is also the active ingredient in the prescription drug disoxin, a stimulant used to treat ADHD, or to help obese patients lose weight. The D isomer is also a metabolic product of the drug bensphetamine, which is a stimulant and appetite suppressant. Thus, the identification of the D isomer of methamphetamine can either indicate the parent drug of disoxin before it is metabolized, or the metabolite of bensphetamine after it has been metabolized. The L isomer of methamphetamine is found in some over-the-counter products, such as Vicks inhaler. The L isomer is also a metabolite of a drug used to treat Parkinson's, called selegiline. Again, because of its similarities to the D isomer, the L isomer affects the nervous system, but because of its differences, it's far less likely to cause a euphoric state or lead to chemical addiction. The DL isomer test differentiates between the two isomers of methamphetamine. Because many tests are incapable of making the distinction between the D and L isomers, A positive methamphetamine test cannot distinguish between the use of the illegal drug meth, the prescription drugs disoxin and benzphetamine, or the over-the-counter decongestant Vicks inhaler. The DL isomer test can isolate some causes of a positive test from others, narrowing down the distinction between legal, non-addictive forms of methamphetamine from the more addictive version. The DL isomer test is reported as a percentage D isomer. Federal drug testing policies have determined that specimens containing 20% or more D-methamphetamine are consistent with exposure to illicit or prescription D-methamphetamine or the prescription drugs disoxin or benzphetamine. These distinctions can offer valuable information to those providing patient care.